This is podcast 33. Okay, in the previous podcast, we introduced meiosis, and we didn't get that far. We only got to prophase 1 and then broke that down more. Today, we'll take you through the end of meiosis. Meiosis is broken into meiosis 1 and meiosis 2. So after prophase 1 of meiosis 1, there's metaphase 1. And in metaphase 1, the bivalents stay together and they line up in the middle of the poles. Okay, as you saw in metaphase of mitosis, this is meiosis. Independent assortment gets set up during metaphase 1. One of the places it gets set up, and we'll talk more about that in a subsequent slide as to what that is. So this is from another book, and during meiosis, these bivalents stay together, and they're held together by proteins. One of the proteins that wraps the bivalents and keeps them together is called sugarshin, and sugarshin is a Japanese word that means guardian spirit. So this is guarding the chromosomes during this phase. Separase that we saw, separase separating the two chromatid chromosomes into one chromatid chromosomes during anaphase, released by the anaphase promoting complex. Separase also works on sugarshin to break this connection, holding this together and allowing the bivalents to separate in anaphase 1 and also in metaphase 2 to anaphase 2 separation, allowing the 2 chromatids per chromosome to go to 1 chromatid per chromosome. So sugarshin is important for holding those together, and separase cuts both the cohesins and other proteins actually that hold the chromosomes together. Again, here's another image from another book, because your book doesn't show it that well. In meiosis 1, you have the chromosomes lining up in metaphase 1, but then they're going to separate in anaphase 1, and that only works if the kinetochore microtubules are attached to the same pole and both sides of the chromosome connect to the same pole. So back here in mitosis, remember, you had a system where in this chromosome that has, in metaphase, that has 2 chromatids, one half of this chromosome is attached to one pole and one half is attached to the other pole, and then after separase cuts the cohesin, so that you get 2 separate chromatids becoming 2 separate chromosomes. They go to opposite poles. In the case of meiosis 1, metaphase 1, both of the kinetochore microtubules for a given chromatid in the for a given chromosome in the bivalent will be attached to both halves of that chromosome, and they both pull in the same direction. So here is a bivalent that's partly getting pulled apart during anaphase 1 of meiosis, and you can see both kinetochore microtubules attached at the centromere of this particular chromosome and this particular chromosome. Eventually, this is going to pull the bivalent apart during anaphase 1. Okay? This does not happen in mitosis, and this does not happen in meiosis 2, where you'd have one of these attached to one pole and the other one attached to the other pole. But in meiosis 1, for a given chromosome, they're both attached to one pole. Okay, so what is independent assortment about? This is a really important idea, because remember, the whole idea behind meiosis is to generate genetic variability in a population, which is the currency of evolution. You have to have genetic variability in a population to have evolution. And you get a lot of variability by that crossing over process. You can see here, this blue chromosome is now going to have red as part of it, and this red chromosome is now going to have blue as part of it, and that didn't happen when meiosis started. So as a result of recombination or crossing over, you have some variation generated. But you also have a lot generated by this process of independent assortment. And a lot of people have some trouble understanding this, and it's not that hard to understand. So if you just slow down a bit or watch this over again, you'll get it. Okay, so let's take this simple set of chromosomes in this diagram. Okay, this set from dad, for this chromosome from dad, this chromosome from dad. So this is, say, homologous pair 1, dad, mom, and homologous pair 2, dad, mom. Same thing over here, homologous pair 1, dad, mom, homologous pair 2, mom, dad. Now, so for this pair here, for number 1, they are lined up left to right from this side of the screen to this side of the screen in the same way. Dad is on the left, mom is on the right. Okay, but for this pair, homologous pair 2, for this cell in metaphase 1, dad is lining up on the left, but for this one, dad is lining up on the right. Well, what's the big deal? Well, it turns out to be a very big deal later on as you process through meiosis. If dad lines up on the left, then after anaphase 1 and telophase and cytokinesis, you end up with 2 cells, okay, as a result of that. And each cell gets 1 of each kind of chromosome. And in this, because of how they lined up here, and the cell is going to split down the middle, this cell is going to get both dad versions of the chromosomes, and this one is going to get both mom versions of the chromosome. Okay? But if they line up a different way during metaphase 1, just this one chromosome lines up a different way, you get different combinations. Okay, here's a dad and a mom a mom and a dad in the cell. And then this is going to split again after anaphase 2 and telophase 2 to make the gametes. And the gametes resulting from this arrangement look like this. Okay, 2 copies of 2 single chromosome, single chromatid copies, both have the dad version of the cell, or of the genes, okay, which is big R at this locus and little y at that locus. Okay, so 2 gametes that both look the same, and both of them have just mom versions of the genes, and or just dad versions of the genes, sorry. And then these just have mom versions of the genes. But just by virtue of this one chromosome pair lining up the other way, so instead of dad on the left, dad is now on the right, after going through meiosis 1 and meiosis 2, the gametes you get are different. So all these gametes have one version from mom and one version from dad, and they have different alleles in them. Okay, little r at that locus instead of big R, and little y at this locus instead of big Y. And the combinations within a gamete, so this gamete has a little r and a little y, okay? None of these gametes have that combination. And this has a big R and a big Y. None of these gametes have that combination. So just by virtue of how you line up during metaphase 1, you'll get completely different combinations of gametes. And this is just one of the chromosome pairs that we're considering in this example. You have 23 pairs in humans, and each of those 23 pairs, dad could be on the right or dad could be on the left. So for any one of those being on the right or left, you'll get a different combination of gametes on the bottom. And so if you consider you have 23 possibilities times 2 being on the right or being on the left, that's a lot of combinations um, of gametes just by virtue of where you line up. And of course, it's not just if number 1 is on the right or the left, if it's number 1 and 2 but not 3, or 1, 2, and 3 but not 4, and so on. So there's many different possible gametes. Just that's without crossing over, just by virtue of where you line up. Do you line up on the left or do you line up on the right? That's called independent assortment. Each of the chromosomes sorts independently. So they sort such that they'll end up with being on the left or being on the right. And that's independent of the other chromosome. Each of this chromosome could end up on the right or the left also. Okay. That affects how genes sort in the final gametes. And then crossing over affects how likely it is when you have one gene, will you also have another gene? So if the two genes that you're talking about, let's say these genes are A and B, if they're far apart on a chromosome, physically far apart on the chromosome, like capital A and capital B in this example, then it's likely they're going to sort independently. So that they're going to sort just like the chromosomes sort. So that if A will be on the right or if A will be on the left, and if big A ends up with big B or not, that's all independent. But if now you have a situation where big A is right next to B, so let's say it's not on either end of this chromosome, but one right underneath another, then because just by virtue of them being really close to each other, it's likely that if a piece breaks off, it's going to take both of them with it. And therefore, if two genes are close to each other, when they form bivalence and there's crossing over, the piece that gets crossed over and pushed onto a new chromosome is likely going to take both of them at the same time just because they're close together. So that's called gene linkage, and there's some famous examples of that. So blonde hair and blue eyes. Those two genes are close to each other on the chromosome. So if you have one, it's likely you also have the other. Red hair and freckles. If you have one, it's likely that you have the other two. It doesn't mean you have to. Doesn't mean if you have red hair, you have to have freckles. Or if you have blue eyes, that you have to have blonde hair. It just means because they are close together on the chromosome, it's likely that if one crosses over to another chromosome, both go at the same time because they're physically next to each other. So if that doesn't happen, okay, that means that there has been crossing over that broke that gene linkage. You had a break, a crossing over break that happened between those two genes. And that's going to be more rare just because the physical space between those two genes is low, is small. And so genes that are close together tend to stay together in the parents, in the gametes. Not in the parents, sorry, in the gametes. Okay. So we talked about how independent assortment gets set up in metaphase 1. All right. So if dad is on the left or dad is on the right, it matters in terms of the final gametes that are being made. But it also matters how you line up in meiosis 2. So in meiosis 2, you have half as many chromosomes, and you don't have bivalence anymore. but you do have double chromatid chromosomes, okay? 2 chromatids per chromosome. And the 2 chromatids in mitosis look exactly the same, because they are exactly the same. But in meiosis, they don't look exactly the same because of crossing over. So let's just say that because of crossing over, this one from mom, this chromosome 2 from mom, has an arm on it that got crossed over from dad. So it's actually blue instead of pink. I added that. That's my version of blue. Okay. In that chromosome where you had crossing over, you're not going to have crossing over at both chromatids. It'll happen on one chromatid or the other. So if that chromatid happens to line up on the left, as it is here, or on the right, as it would be in another situation, it'll depend on where that gamete ends up. So here it ends up on lining up on the left, and so it ends up with the same. It ends up with in a gamete with another chromatid that was lined up on the left. Okay, and the one that is on the right does not have that same chromatid in it because it didn't have crossing over happen. So again, how these chromosomes line up during meiosis 2 also affects it because of the changes that happen during crossing over. So during crossing over, what these chromosomes look like is going to change. And what they look like will not be the same on both chromatids. And therefore, if the chromatid lines up on the right or the chromatid lines up on the left during metaphase 2, also affects the final gametes that are made. So this is yet another way of how independent assortment can affect variability. So you get variability from crossing over, exchanging bits of genetic information among homologous chromosomes during synapsis in prophase 1. You have variability created by how the bivalents line up during metaphase 1, if they line up on the left or the right. And then you also get variability from how they line up in meiosis 2 in metaphase 2, if the single chromatid with the change in it that is part of a 2 chromatid chromosome lines up on the left or lines up on the right. It matters in terms of which gamete it's going to end up in. Okay, so now we've gotten past prophase 1 and metaphase 1. In anaphase 1, the guardian spirit Shugashin is going to keep those bivalents together. And when that bivalent gets pulled to one pole, they're going to have 2 sets of kinetochore microtubules attaching to a single bivalent and pulling them in one direction. That happens in anaphase 1. And then there's going to be telophase 1 and cytokinesis, and a very brief interphase between meiosis 1 and meiosis 2. They go right from meiosis 1 into meiosis 2 with a very brief pause. Sometimes the nuclear envelope will reform during that after cytokinesis of meiosis 1 and before meiosis 2 starts, but sometimes that doesn't happen because it's so brief. The chromosomes stay mostly condensed, but you'll see in the video at the end they sort of recondense going into meiosis 2. And a big thing I want you to know is if we look at the number of chromosomes per cell, 1, 2, 1, 2, it's going to be 2 different cells, okay? That is half the number that we started with, okay? So here's one cell that has 1, 2, 3, 4 chromosomes in it, and after the end of meiosis 1, after cytokinesis of meiosis 1, you'll have 2 chromosomes per cell in this example. So whatever you started with in meiosis 1, after meiosis 1, you already have gone to haploid. So you start off with a diploid cell in meiosis 1, and at the end of meiosis 1, you're already in haploid. You're not done with meiosis, but you've already gone from diploid to haploid after meiosis 1. Okay? In meiosis 2 is just like mitosis. Okay? Meiosis 1 is very different from mitosis, but meiosis 2 is just like mitosis. So, in these two cells, in both of them, during prophase 2, you'll see again the nuclear envelope will disintegrate if it reformed at all. You'll see these centrioles moving to opposite poles and the spindle getting set up. Okay. You'll see kinetochore microtubules attached to the centromeres of these chromosomes, 2 chromatid chromosomes. You've got one attached to one side, you've got the other one attached to the other side, to the other pole. You have spindle microtubules set up, and then the chromosomes line up in the middle of the cell during metaphase 2. Separase is going to cut the cohesins that is holding these 2 chromatids together in one chromosome, and they'll become 2 separate chromosomes that get pulled to opposite poles during anaphase 2. And then you have telophase 2 and cytokinesis, where you go from 2 cells to 4 cells. Each one has haploid daughter cells, half the number of chromosomes you started with, 2 chromosomes per cell. Each cell has a single chromatid. So meiosis, you started with 1 cell, and at the end of meiosis, you have 4 cells. In meiosis, you start with a diploid cell, and you end up with 4 haploid cells. Okay, let's all bring it all together here in this YouTube video. Now, if you want to look up and use the link for the whole video, there's a good introduction here in the first 2 minutes about what meiosis is for and what it's about. But this has some really nice animations on what is happening during the different phases of meiosis. So I'm just going to play that now.
Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over.
This happened in
After crossing
1.
over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the 2 centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs.
So she's talking about they line up randomly, different combinations. That's independent assortment. So if this guy that is part red, part blue, lined up on the left instead of the right, you'd end up with different gamete combinations at the end of the experiment. And notice how during anaphase, when these separate, the bivalents are going to separate, but the new pieces of DNA that got exchanged stay with the new chromosomes.
The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the 2 poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis II.
Okay, so at this point, each one of these cells is haploid. The cell we started with was diploid. So after meiosis 1 is complete, you've already gone from diploid to haploid.
Meiosis II is similar to mitosis. The first stage of meiosis II is prophase II. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase II and prophase I is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase II, there is no synapsis of homologous chromosomes or crossing over. In metaphase II, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align.
So again, this is saying, this is another aspect of independent assortment is during metaphase 2, how the cell lines up those chromosomes. If, for example, this funky chromosome that's partly red, partly blue happens to be on the bottom instead of the top, you'll get different combinations of gametes.
In anaphase II, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase II, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis II began with two cells, and each of those cells were split into two cells, we now have four unique haploid cells at the end of meiosis. These cells are gametes. Two gametes, one from a father and one from a mother, may fuse to produce a diploid embryo.
Okay, so that's a good, simple summary of meiosis, bringing together meiosis 1 and meiosis 2. We went through them slowly, but that's putting it all together at the end. Hopefully that made sense to you. And here's an H5P question for you.